Hello, and welcome to our webinar, Rapid Sterility Testing, Enhancing Compliance, Efficiency, and Accuracy, presented by American Pharmaceutical Review magazine and sponsored by Nelson Labs. My name is Mike Auerbach. I'm the editor-in-chief of American Pharmaceutical Review magazine, and I'll be the moderator during today's presentation. This webinar will examine how rapid sterility testing technologies enhance efficiency, reduce time to results, and improve overall product safety while maintaining regulatory compliance. The webinar will also review a case study to gain further insight into the accuracy of rapid sterility testing solutions. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note, the right side of your screen also features an overview of this webinar and more information regarding our speaker. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. Our speaker today is Nina Moreno. Nina is a certified microbiologist and global segment director for sterility assurance at Nelson Labs. She has been with the company for over 20 years and has experience providing technical and strategic oversight for microbiological laboratories. Nina is a member of the Kilmer Microbiological Quality Process Analytical Technology Team and the Modern Microbial Methods Collaboration Group. Nina, we look forward to your presentation.
Hi, everyone. Thank you for joining today to discuss rapid sterility testing, enhancing compliance, efficiency, and accuracy. So I know we just heard this information, but I also wanted to mention that I started out my career with Nelson Laboratories in our sterility testing suite in the clean room. So sterility testing is near and dear to my heart. For the agenda today, we'll go through some background information around regulatory requirements and expectations. We'll do an overview of what an ATP-based system is and how it functions. We'll also review data from a case study, and then we'll go through a summary and Q&A. Okay, so first things first, we want to make sure that we have our terminology straight. There are many different ways to refer to rapid and alternative methods. So you can see there's rapid microbiological methods, alternative microbiological methods, modern microbiological methods, and rapid microbial tests. And essentially, all of these acronyms are really pointing to test options that are the non-referee test. And so that's basically been something other than something like USP 71. In the general guidance for alternative methods, this can be found in USP Chapter 1223, Validation of Alternative Microbiological Methods. And I think it's important to point out a few things that are outlined in this chapter. So the first is it states that the actual limits of detection of compendial microbiological methods have never been established quantitatively. And so while we all might feel very comfortable with the compendial, the referee method, the limits of detection haven't really ever actually been established. It does have a very long history of use, though. The second item that's pointed out here in chapter 1223 is that the CFU, as an estimate with a variety of limitations, is prone to underestimate the number of cells. And that's due to the way that cellular growth takes place versus how the CFU forms. So CFU might be made up of a clump of viable cells, and so, again, while we are very comfortable with a CFU in general, because of its long history of use, there are some limitations that are associated with that form of measurement. The next thing to point out is that USP has long provided support for the implementation of alternative assay methods, and that's noted in these general notices. And so, while there may be concern and a perception that regulatory agencies may not look favorably upon an alternative method, in fact, there's a great deal of support, and it states that in the USP. The next thing to point out is that there are guardrails in place. And so while there may be questions about an alternative method and the validation process, these guardrails really help to make sure that the methods that are being used are adequate and address the questions around potential for contamination and potential for patient risk. So all methods and procedures in USP that are below 1,000 are considered the referee tests. And then in addition to that, there are harmonized compendial methods. And really the expectation with an alternative method is that that is approached by region and that there's a good understanding of what the requirements are for the alternative methods in each region that might be applicable for your product. And here's a very long list of other useful references. And again, these are very useful, makes for good weekend reading if you'd like to dive in. But a couple of things I wanted to highlight here is that USP Chapter 1071, the RMM for short life products, will have an update on the 1st of August of this year, 2025. And then there will be two new chapters coming out, which is a huge update in terms of compendial processes and what we're able to utilize for testing purposes. So the new chapters will be USP 72 and 73. 72 is for respiration based method, and then chapter 73 is for the ATP based method for detection of contamination. And both of those will also go effective the 1st of August, 2025. And those are essentially tied to chapter 1071. And since this is such big, exciting news, I thought it would be helpful to dive into USP 73 and what's outlined there. And I'm having a focus on ATP based methods for our discussion today. So, USP 73, which again is for USP, it is ATP based method. In the introduction, it states that this chapter is describing a risk based test for the detection of microbial contamination, and it is specific to short life products. And that includes items that have short shelf life in terms of expiration, products with short manufacturing times, and then any kind of in process control for product intermediates, cell media, process solutions, other items that might really benefit from having a faster time to result. And then this last statement is also going to be found in USP 73 when that becomes effective. And that's that ATP based testing has a long history in pharmaceutical, over the counter, and cosmetic industries. And so while there may be some general hesitation when it comes to alternative methods, the process itself has a very long established history, has been used effectively to address the potential for patient risk. Okay, in chapter 73, there are pros and cons that are outlined directly in the chapter. And so the first, the advantages, is that there's a broad equivalency to the compendial methods. So it's very easy to compare results from the compendial to what you might achieve with an ATP based approach. The second is that there's early detection of microbial growth. Of course, that's the main reason that the products that have been approved seek this type of a process is to have this early detection opportunity. And the third is that you can still isolate organisms for identification. And so this is very important if you're looking to understand potential sources of contamination, if you need this information for trending and investigation. This type of a process, this ATP based process, does allow for organism identification. There are many other types of alternative and rapid methods that are available, and some don't allow for this. Okay, so for the disadvantages that are outlined in this chapter, there are two. So the first is the inability to detect organisms that do not grow under the conditions utilized. And you might be thinking to yourself, doesn't that apply to most microbial tests? And yes, that would be the case. So even for something like USP 71, which has that long history and that established use, uh, even with a test like that, you're only going to be able to detect organisms that are able to grow under the conditions utilized. So, as an example, you likely wouldn't detect geobacillus because of the temperature conditions that are required for that organism. Okay, the second disadvantage is that products with significant ATP background that cannot be reduced wouldn't be able to use this method. So the second disadvantage is really just pointing out that unfortunately you might not be able to use this approach for all products that are out there. So not necessarily a disadvantage of the method, but maybe a potential disadvantage in that it can't be used for everything if your product does have its own ATP background that can't be um, reduced. So, overall, when we're looking at the regulatory landscape and how you're able to comply with expectations, there's certainly support for the use of alternative and rapid methods globally. And with these new chapters that are coming up, it is essentially doubling down that we should have compendial methods in place for the use of these alternative methods. Okay, so I'm going to go into an overview of the rapid sterility test process using an ATP based method. But before we get into that, I want to make sure that we're all on the same page when it comes to sterility testing in general. So, this process utilizes either a membrane filtration approach or a direct inoculation, where the product is placed directly into the test broth or run through a filter, and then the filter is tested in broth, and then the containers are inspected for signs of growth visually. So this can look like turbidity, maybe flocculent growth. There could be a sediment down at the bottom of the container of media. And so that's generally how the test is performed. Now, in some cases, there are products that might make the media look murky, or there might be um, uh, uh, ingredients that are utilized as a neutralizer to cause the media to look cloudy and turbid, even when there's no growth present. And in those cases, the test would require a subculture or a media transfer, and that involves taking a 1 ml aliquot from the original broth, placing that into a fresh container of media, and then incubating that for an additional 4 days. So original incubation, 14, and then for those products that require media transfer subculture, an additional 4 days of incubation. So it's quite a long process. Now, when we're looking at the Celsius Advanced 2 system, it's similar in that the sterility test is performed in exactly the same manner. So either in a clean room setting or in an isolator. The item is membrane filtered or placed directly into broth, and then those broth containers are placed into incubation. Uh, but this is where the differences will become apparent. So rather than a full 14-day incubation, we're able to shorten that. And the reason that we're able to do that is because of the fact that all living cells have energy that is stored as ATP. And when there are viable cells present, ATP will be present as part of the metabolic process. And because it's present, we're able to utilize reagents and equipment to detect the presence of light using the luciferase enzyme. And these kind of elevated readings of light, so this RLU measurement, the relative light units, will be detected through the equipment that you see there on the right. And this reaction that takes place utilizing the ATP and the luciferase enzyme allows for much earlier detection of growth than what you could see visibly. And so the way that I like to describe this is with fish. Now, I'm not much of a fisherman, but I can probably scoop a goldfish out of a bowl. Now, the way that this process works is this type of a detection, rather than trying to find just one single cell that's present, um, there's an enrichment phase that goes along with this ATP based method. I mentioned we'll do the testing, samples will go into the incubator, and that process is typically about 6 days for the enrichment. So that enrichment allows for additional growth of cells to increase the presence of ATP that's available. Because we are taking a small aliquot from that broth container and then placing that into a cuvette that's then analyzed for the measurement of relative light units. Now, we're able to detect this at a much lower level because there's an amplification process for this ATP. So you go from having the equivalent of one goldfish present to a few goldfish that are actively there. And then this amplification allows for much increased detection, like what you would see here on the right, that's just filled with fish. So it's very simple and consistent to be able to detect the presence of ATP after a very short period of time. So let's take a look at USP section 1071. Currently, the chapter states that ATP bioluminescence is a well established technology, and there are approximately 5,000 relative light units for the equivalent of 10 to the third CFU. And really, with an ATP based measurement, you can detect the presence of organisms 3 to 4 logs less than what is required for that traditional visual inspection where you're looking for signs of turbidity or growth in the broth. Um, and of course, there's that need for that threshold level of ATP to have that consistent, accurate measurement. I wanted to point out a couple of changes that are going to be part of the update that's going live on the 1st of August, 2025. So the first is that if the RMM is not described in a general chapter, then USP 1223 is required. And so that's been the approach up until now because we haven't had these other compendial methods available for USP 72 and USP 73. The second thing it states that if the RMM is described in a general chapter, then primary validation and respective USP chapter apply. So essentially, you don't have to follow all of the requirements that are outlined in USP 1223. It's a much more straightforward approach to be able to utilize a rapid method. So the effect of all of this and the ability to measure ATP in relative light units is that you can go from a test, a sterility test that might have a 14-day incubation under USP 71 down to 6 days. Or if you are in the category where your test requires that media transfer or the subculture, you could be going from an 18 to 20-day incubation all the way down to 6 days. So, certainly a much better time to result. And if you're in the case where you have to administer the product to a patient prior to 14 days, anytime that you can remove from that overall time to final result can be really helpful. In USB 73, there are a couple of items of note that I wanted to point out since it is a very different approach than what's outlined in USP 1223. That is that you will use the normal method suitability organisms that we're familiar with from USP 71 chapter and a selection of organisms relevant to your product or your manufacturing process. So that's very similar to what we already experienced with USP 1223. But the difference is that slow growing organisms and local isolates may be included if relevant for your process or your product risk. And so you wouldn't necessarily have to include a slow growing organism like C. acnes unless it were relevant to your process. So that's a pretty significant change. The inoculum level for your method suitability would need to be less than 10. So, again, that's a difference where we're accustomed to having not more than 100 CFU for the method suitability test. You do need to perform a sample interference study, and that is to show that the equipment, the reagents, the process are all effective for accurately measuring the presence of contamination, and that you're not getting interference, or like was mentioned in the disadvantage, high background ATP levels that can't be reduced. And the incubation time that's required per USP 73, rather than being a set time frame like what is listed in USP 71, for USP 73, it's dependent on the time it takes to detect the relevant organisms, plus a safety margin in terms of generation time. And so this example calculation that you see here on the screen is directly pulled from USP 73. So let's imagine that you have a panel of organisms already established. You've determined that slow growing organisms aren't relevant. And in fact, the slowest growing organism, the time to detection is maybe 48 hours. So if you go through this calculation, it shows that really your time to result in this type of a situation could potentially be just over 2 days, which is uh, incredible when the standard test that we look to in USP 71 is a 14 or 18 to 20-day test. Okay, so now that we understand the regulatory landscape and we understand how the ATP based system works, let's take a look at our case study. So, this was part of the performance qualification that we performed at Nelson Labs. And there are several factors that we looked at because we were doing this under USP 1223. And so, those factors include limit of detection, equivalency, specificity. So, there's a number of factors that we need to analyze and address to make sure that the method is suitable. As part of the method, we use the organisms that are outlined in USP 71, so that panel of 6 organisms for the method suitability at multiple levels of titer. We also utilized 3 environmental organisms, again at varying levels for the titer. We did use a slow growing organism since that was the expectation looking at USP 1223. We included stressed organisms, of course, negative and positive controls, and then did a side by side analysis with the compendial test, and then performed a statistical analysis to show that the method for this ATP based process was non inferior to the compendial method. So that's the approach that we took. When we look at the results, we found some interesting things. They're definitely worth some discussion. So here are the organisms that we utilized. I've highlighted the use of C. acnes because, again, that's the slow growing organism. And in many cases has been a limiting factor for the way that the test is performed for alternative methods and the time that's required for the test. So we used all of these organisms, and at 6 days, we pulled an aliquot, ran that through our Celsus system, and then also viewed the containers visually to look for signs of growth at 6 days of incubation. Now, with C. acnes, we had some interesting results. Looking here, you can see one of these things is definitely not like the others. So that was a pretty easy score there, that this container was positive for growth. Some of these containers were also very clearly negative for growth. And so you can see that there, that those 3 containers were negative. But we did have one that was kind of a question mark. And it wasn't really clear whether there was growth present or not. And so if I zoom in here for that up close picture, you can see that in the broth, there are some type of like a particulates, debris, could be interpreted as early growth or turbidity, something that did not look like the other containers, which were completely clear. Okay, so let's do a deep dive into these results. So we had 6 different replicates for our C. acnes containers. And again, keep in mind, these are slow growing organisms. And so, um, you know, this is something that can take a bit of time to be able to show up in the compendial method. We utilized anaerobic media, so this is our fluid thioglycolate media. Inoculation levels were very low, so approximately 1 CFU. And so that means that containers may or may not show up as positive for growth just due to distribution of this very low level of titer. Okay, so going through our columns, we have our 6 replicates, our 6-day visual. So this is us looking at the broth container and inspecting for signs of growth visually, like turbidity. We have our rapid sterility results. We also did a 7-day visual score since that's approximately halfway through the typical compendial incubation time. We performed a subculture on agar plates. We did a 14-day visual and then a 14-day Celsius. So many results that were all compared uh, as part of the assessment. Okay, so to address this question mark that we have, let's take a look at our replicate one, which is the one that we had a question on. Visually, because it had this debris or something that was going on in the media, we did mark that as positive for growth because it wasn't clear like some of the other containers had been at day 6. Now you can see the rapid sterility results that we obtained at day 6 shows that this replicate 1 was negative for growth. When we look at replicate 2, we also had a discrepancy between what we were seeing visually versus what we were measuring through the use of the Celsius. Visually, it looked like it was very negative, clear, nothing in the media that could indicate growth. But the rapid sterility results showed that it was, in fact, positive for growth. And then we have a few results where the results were in line for the different replicates. And then for replicate 6, same as replicate 2, where we had that discrepancy. Okay, so let's take a look at day 7. So we did have a difference in the visual results between day 6 and day 7. Um, so that shows as still positive for growth visually. Again, didn't see a lot of change, but there were starting to be questions around whether this was actually growth or whether there was something else going on in the media container. But the rapid sterility results again showed that it was negative for growth at day 6. When we did the subculture to agar plates, you'll see that all of the results matched exactly what we had at day 6 for our rapid sterility test results. So no differences there. For the Celsius 14-day results, again, that matches perfectly with what we saw in the Celsius at day 6. And we did have to perform a media transfer for replicate 1 just because it hadn't really changed over the course of these 14 days of incubation. And so it wasn't clear that it was really growing. There might be something else going on in the media container. So we did do that subculture media transfer process that requires the additional 4 days of incubation. And we found that it was, in fact, negative for growth after following that process. So we were able to go back and say, okay, that the actual final result for this replicate 1 is that it was negative for growth. So the important thing to note here is that for these test samples, there were no discrepancies between the 14-day visual and the 6-day rapid sterility test results. Those were completely in alignment. But we had to wait a significant number of additional days in order to get to the same result. And when we're talking about the media transfer, of course, that's another 4 days. So ultimately, we had to wait 18 days to get to the same thing that we already knew based on the ATP based results at day 6. So really, the method that's used for ATP is very effective and highly accurate. Because of this measurement of the ATP and its comparison to background levels of what might be present in your media container, you can really take this very long testing process, potentially 18 to 20 days, limit it down and have it be a very objective, instrument-based result that you can have consistent, accurate information very early on in the process. Now, at this point, you might be wondering about whether this type of a rapid sterility test is applicable to your product. And the nice thing about the Celsus system is that it can be used for items that are tested through direct inoculation. It can be used for devices, items that are filtered, and many other systems that are available require the use of a filtration step. But because of the way that this test system operates, and it's very consistent to what you would see in USP 71 in terms of how the test itself is performed, because of that, it has really broad applicability. And essentially, we would just need to make sure that we don't have those high background levels of ATP that are coming directly off of the product. But really, it can be used for most items that are uh, currently being tested using USP 71. So, in summary, this type of an approach is highly compliant, utilizes established technology that has been reviewed many times by the FDA and other regulatory agencies, and it's a validated method. So, intense validation that goes along with an alternative approach. This type of an approach is also very efficient, so we can understand correct, accurate results much earlier in the process than we can with a compendial test. And because it is objective, it's an instrument-based result, we can avoid some of the questions that might come along with situations like media transfer or subculture, and then conflicting information from what you might be able to observe visually. So overall, the system is fantastic and accepted and reliable for your sterility testing needs. Now, if you are interested in additional information, you can get that here. So this QR code will take you to the Nelson Labs website. And if you'd like to submit a form to request more information, we're happy to have a conversation with you uh, for your specific test and product needs. And with that, we'll go over to Mike for questions. Thank you.
Thanks, Nina, for your excellent presentation. I'd like to remind everyone that you can submit any questions you have for Nina into the Q&A box on the screen, and we'll get to as many as we can. So we do have a couple of questions in. Nina, our first question is, how many samples are required for testing?
Oh, yes. Okay. So that's a great question because there are going to be some changing requirements. So, in the current revision of USP 1071, it's dependent on the total volume of product that's been produced. And so it's a bit more straightforward in the current revision of 1071. And the current revision also makes reference to the European Pharmacopoeia. Uh, and so the way that that works is if you have between 10 and 1,000 milliliters of total volume of product, then you would test 1% of the volume. And the nice thing about that is it takes an approach of understanding that there may be limited volume that's available for testing small volumes that are being manufactured. And so, that type of an approach does make a lot of sense. Now, in the upcoming USP 73, it basically says that if you are manufacturing into multiple containers, then the way that the sampling would take place is you would follow USP 71 guidelines. Um, and so that might be a challenge. And so it does also state that if you have low product volume available, then you can work with your local regulatory agency to determine a best approach for the sampling method. So it's going to be a little bit different in USP 73. Basically says follow 71 unless you have these lower volumes, and then work that out with your local regulatory agency.
Excellent. Thanks, Nina. All right, let's move on. We have a few more questions that are already in. Our next question is with ATP based rapid testing, how does this method distinguish viable microorganisms from non viable microorganisms?
Okay, yeah. So this is a great advantage of an ATP based approach because some of the questions that may come up is, am I going to get more hits, more recoveries using an ATP based system than I would if I were using a compendial approach? And because an ATP based system is specifically based on an enzyme approach combined with this reading of a light in relative light units, uh, it's not going to be picking up things that are non-viable because ATP is associated with the presence of living organisms, and so you shouldn't be picking up non-viables with an ATP based approach. And in 1071. The new revision that's coming out on the 1st of August, it will be stating that the amount of ATP that's present and the light that's being measured and emitted is going to be in proportion to the amount of ATP that's present. So if you have more organism there, you're going to see more light. But if you have things that are non-viable, it's not going to generate any ATP, which therefore will not be amplified and not picked up in the system. So this method is really great for distinguishing and picking up specifically viable organisms and not the non-viable.
Mhm. Excellent. Thank you. Another ATP based system question here. Is there a maximum media volume that the system can handle?
Yeah. Again, a really important question because there are devices or sometimes like a liquid material that has to be diluted down. And so larger volumes of media are needed for testing. And uh, the lowest volume that we utilize here at Nelson Labs is 180 milliliters, is kind of on the low end. And so we're able to readily pick up growth at that volume. We also use much higher volumes again because we provide testing for many different types of devices and other products, and so we do utilize quite large volumes as well. Sometimes around 2,000 milliliters, 2,500 milliliters, and we're able to detect growth in these larger volumes as well within the same time frame. So again, a really flexible system and really good accuracy, even at these higher volumes that might be needed.
All right. Excellent. Just a reminder, if you have any questions for Nina, to type them in. We'll get to as many as we can. Our next question is, what was the most challenging part of bringing up a rapid method?
Yeah, so I know this can sound the whole approach might sound intimidating. I think what was the most challenging for us is maybe all of the upfront work in assessing, okay, what kind of an approach do we want to use? What type of equipment is going to make the most sense? So this upfront assessment for user requirements was pretty in depth. So that part took quite a bit of work, and then there's always getting the funding for the purchasing, and so going through that whole process. In addition to that, maybe sometimes it can be hard with utilizing environmental organisms to have those grow in the way that you need to support this testing, because organisms are living and they're gonna do their thing, and so that sometimes can be a challenge, but that's something I think that's being taken into consideration with the upcoming chapters, because it does allow for the use of um, purchased organisms that are similar to what you would find in your environment. Again, knowing that how challenging it can be to propagate the organisms from the environment, you can use something similar that's available for purchase in place of that.
Right. Excellent. All right, let's go on to our next questions. I'm going to combine these next two ones. What other organisms showed any discrepancies, and how were the organisms stressed in the study?
Yeah, so we didn't see a lot of discrepancies in the testing, which is good. That's what you want to see. And again, with what we saw with the C acnes, it was a discrepancy until we got to that final result. And so we still did end in the right um, place for the organisms. So not a lot of discrepancies, but in the way that the organisms were stressed is through heat. And so these organisms all went through a heating process, and then into ice bath, and then testing from there. And we needed to know that there was a 2 log reduction in the amount of organism in the population. So that way we can prove that it was actually stressed. So that's how we approach that.
Terrific. Thanks again. Moving on, we've got our next question, which is, what are the challenges faced in validation of testing of solution versus testing of suspension?
Yeah. Good question. I don't know that there's a specific challenge for one versus the other because both, a solution and something that's a suspension can be tested in using this approach. Um, Suspensions I know can be challenging with the compendial method because of the way that it might appear in the media, leading to subculturing and media transfers. There aren't really any specific challenges that I've noted between these different types of products, which is good. Part of the advantage of using this type of approach rather than a visual check for turbidity, as long as we're able to go through that sample effect testing to make sure that there's no interaction from the product with the reagents or the test system, then we can be able to proceed.
Got it. Thank you. Just another reminder, if you have any questions for Nina, to type them in. And as I mentioned before, we'll get to as many as we can. Our next question is, are either the Celsius Advanced 2 or the Celsius Amp Screen suitable for testing cell therapy products?
Oh, yeah, yeah, great question. The Celsus system, is suitable for, cell therapy products. Again, I know there can be challenges because of the material that's in there and potentially causing some, background noise, but we have a way to be able to reduce and suppress, that background noise and focus on what might be coming from, the actual product itself and from organism that could potentially be present. So the Celsus works great for cell therapy type of products. There's also, by the way, I don't own stock in Celsus or anything like that. It's just a great product that we've really appreciated being able to work with. For cell therapy products, there's also the ability to use kind of a piece of equipment that goes along with it that's called the Adapt Concentrator to help, again, reduce some of the potential impact that could come from materials that are in cell therapy based type products.
Got it. Thank you. All right, we have another question here on stressed organisms. The question is, as part of the validation work, you mentioned you tested stressed organisms. Can you describe conditions to stress them? Were they exposed to a sterilization process?
Oh, yeah. Okay, great question. For the purposes of this study for supporting rapid sterility testing, these organisms did not go through a sterilization process as part of the stressing. It did go through a process where we brought the organism up from a frozen state, then allowed for that to grow out and then did the heat stressing approach. And so that's how the organisms were stressed is using heat. This is a good kind of broad approach for the different panel of organisms that need to be used in a 1223 approach, and so that's what we use. But I can see how there could be benefits if your specific sterile process requires a sterilization method, and then utilizing that sterilization method as part of the stress. I can see how that would be um, a reasonable thing to do.
Mhm. Excellent. Thanks again. All right, let's go on to our next question. For the 6-day sterility test, what are the requirements you recommend?
Okay, so if we're looking at the 1223 type of approach, then that's where we would, uh, you know, want to look at making sure that you have organism available from your process, from your environment that can be utilized for the testing. Uh, we want to go through and validate the method's suitability. So making sure that there's no interference from the product and the test system and sample effects testing. And really, that's what's needed is making sure you have the organism available, that you go through this method suitability and sample effects testing, and that is what's involved with getting to the 6 day test.
Got it. Excellent. Thank you very much. All right. Our next question is this method still compliant for use with ISO 11137 sterilization validation?
Yeah, so this is definitely a question that I've heard come up before, and the great thing about using this type of approach is it's also supported through ISO. So I know I've focused a lot on USP and because of the 1223 approach and the changes that are coming. But the way that ISO 10137 is set up is it does also allow for alternative methods. And so, yes, this type of an approach can also be used to support this type of a sterilization validation.
Mhm. Excellent. Thanks again. All right. We still have a few more questions to get to. And our next question is: did you investigate what made the negative canister look like there was growth?
Mhm. Yeah, so, interestingly enough, this kind of thing does happen in sterility testing, from time to time. You might see, something like what I displayed. There might be sometimes these kind of like darker colored flecks or things that can appear in the media. We do have the ability to help, go down this path of detecting what might actually be present in the test container. For the specific purposes of this test, we didn't do that, but we have um, certainly options here at the lab to be able to support something like that.
Mhm. Excellent. All right. Let's go on to our next question. Um, if the product has ATP, is there an ATP reduction method?
Yeah. Okay. So, yeah, this is going to be important if you do have a product that is actively contributing to the presence of ATP. What we've used is a reagent kit available through Charles River that helps to suppress this. Um, ATP that might be coming directly from the product. And I know I had mentioned the use of the Adapt Cell concentrator. So these are kind of some options that we can use to uh, try this process with a product that might have higher levels of ATP to make sure that we're not getting interference from what might be inherently present in the product.
Okay. Just one more reminder, if you have any questions for Nina, to type them in. Let's look at our next question. So our next question is: for sterilization validation purposes, I assume that method suitability would still be conducted the same way, compendial, or would you be able to use the system?
Oh, yeah. Okay. So yes, we would still conduct the method suitability in a similar manner. We would also incorporate the use of these other organisms. So that's where there's some differences because we're not, just using the panel that's typically used for the test. We would also want to incorporate the use of um, environmental isolates or something that could be relevant and important for your manufacturing process. But yes, it could be conducted in a similar way.
Excellent. Well, I believe that's all of the questions. Nina, thank you so much for all your time today, and thank you for answering all the questions. In addition to thanking Nina, we'd like to thank Nelson Labs for sponsoring today's event. I would like to remind everyone to keep a lookout for an email containing a link to view this webinar on demand and to allow you to share it with your colleagues. Thank you, everyone, for attending today's webinar, and please enjoy the rest of your day.